Welcome to AP Biology and Lecture 1.5a. Today we're going to be talking about carbon and the role of and its role in macromolecules. Okay, and today we're going to specifically be focusing on one group of biological macromolecules, and out of the four that there are. Okay, so my four categories are carbohydrates, lipids, proteins, and nucleic acids. And in lecture 1.5a, we will focus on carbohydrates, and on lecture 1.5b, it will be on lipids. Okay, both carbs and fats do provide energy for our bodies that allow us to fuel all of the activities that we do, whether it's running, swimming, sleeping, just basic maintenance. We need to have fuel, and that fuel comes from the most part from carbohydrates. Okay, and then of course we do get some extra fuel from fat. So we're going to focus on carbohydrates, like I said. And one thing that all carbohydrates have in common is that they contain three elements: carbon, hydrogen, and oxygen. What I like to say is CHO, carbon, hydrogen, oxygen. Okay, and they contain that carbon, hydrogen, oxygen in a 1 to 2 to 1 molar ratio. For example, glucose is C6H12O6. And if you simplify that, that is a 1 to 2 to 1 ratio. The empirical formula is CH2 to the O, as you can see, the numbers of hydrogen is doubled. And that little N subscript there, that stands for the number of carbons. So if I had 12 carbons, what would be my formula? It would be C12H24O12. Okay, and carbohydrates basically can be classified into one of three groups: mono, which is a single carbohydrate; di, which is two carbohydrates put together, such as table sugar, which is sucrose. An example of monosaccharide would be glucose. And then polysaccharides, which are many sugar units stuck together. And examples of that would be cellulose, as I mentioned, with wood, or starch is also a polysaccharide. So these are some examples of monosaccharides, and the formula for the 3-carbon sugar would be C3H6O3. What would it be for ribose and deoxyribose, which are both 5-carbon sugars? Okay. And then we have our 6-carbon sugars, and as you can see, they will all have the same empirical formula. All right, I want you to write what that empirical formula is on a question. You're going to have to figure that one out. Okay. Again, carbohydrates can be linear or they can be ring-shaped. And generally, a linear carbohydrate thrown into water will turn into a ring. There's an aldose. Okay, so sugar comes in many different isomers. As you can see, when we look at our hexose sugars over here with the formula of C6H12O6, they have the exact same formula, but notice the arrangement of the atoms. And right here, the structural groups are arranged slightly different. And remember, structure determines function. And so that actually changes what type of sugar it is. Fructose is also C6H12O6. Okay, and again, we see a different arrangement. It has different properties. Ribose and ribulose are both 5-carbon sugars with the same formula, C5H10O5, arranged differently. They have different properties, and the same thing with the triose sugars, C3H6O3. Structure determines function. Okay, glucose is probably one of the most important monosaccharides that we have out there. And again, it is the raw material for the synthesis of other monomers. It is oftentimes what our mitochondria are breaking down and turning into ATP. And so you must know that one. And generally, whenever you see this sort of 6-sided shape, that represents a glucose. Now, what you notice here is we can see all the carbons in this linear form, and you can see the carbons in this ring-shaped form. But what you don't see, and you can see the carbons are all labeled here, but in this particular model, you do not see the carbons labeled, and we assume there's a carbon at all of these points. Okay, there's a carbon there, there's a carbon there, carbon there, carbon there, carbon there, and there's my 6th carbon over there. There is an oxygen right here, and that is put in there. Okay, so you have to become familiar with these types of models, and do remember, if you see a shape, a hexagram, okay, with 6 sides, then that is a glucose molecule. It represents a glucose molecule. Disaccharides consist of 2 sugars that are put together. Sometimes those sugars can be the same, like a glucose and a glucose makes a maltose. A glucose and a fructose makes sucrose, and sucrose is table sugar. A galactose and a glucose makes lactose, which is milk sugar. Okay, and they are joined together right here. And this is this is also a 6 carbon sugar in a 5 carbon format. There's my 6 carbon up there. Okay, and these are 2 hexagon shaped 6 carbon sugars. Okay, so what would be the formula for a simple disaccharide? Remember, it's C6H12O6. So if I put 2 of those together, what's my formula going to be? Well, if you thought the formula was C12H24O12, you were wrong. Why? Because remember, we have to put together these monosaccharides via dehydration synthesis, which means we have to remove a water. Dehydration means to take a water. Synthesis means to build. So where does that water molecule come from? It comes from a hydrogen here and an OH, a hydroxyl group there. There's my water coming out, and we form that linkage. Now, this just doesn't happen on its own. There are a whole bunch of different types of enzymes, which we'll discuss later, that to do this, and it does require an input of energy. Excuse me. Polysaccharides, they are multi-purpose. They have lots of different roles. Both animals and plants have polysaccharides. Okay, plants, it's called amylopectin. You might have heard of pectin if you do any type of canning, like making jams and jellies and things of that nature. Pectin is what helps to solidify that jelly so it's not just a runny sauce, and sometimes you have to add pectin into your jam when you're making it. And that is found only in animals. It's not very branched. It's a very large, loosely packed molecule. Glycogen, however, is a storage form of sugars in animal cells. And glycogen is highly branched. It can be packed in more tightly into animal cells. We don't have very much of a supply of glycogen in our cells. Our liver actually stores most of it, and it can be broken down and utilized very quickly by a hormone that is released by your hypothalamus called glucagon. Well, actually, hypothalamus signals release the hormone, which then is released from your pancreas, and that hormone is called glucagon, which travels through your bloodstream to the liver, breaks down the glycogen, which is a polysaccharide, into monosaccharides, which then go into your bloodstream and then travel to the mitochondria, various cells all over your body, to the mitochondria, where they are broken down and turned into the energy form that your body can use, which is ATP. Okay, and so again, polysaccharides play a main role in energy storage, glycogen in animals, starch in plants. They can also play structural roles. In plants, it's going to be cellulose. That's what basically makes up wood fibers. It's what makes up the cell walls of plants. In animals, it's in something called chitin, and that is found in the exoskeletons of insects. It's found in the exoskeletons of things like lobsters and crabs and things like that. Okay, and they're actually put together slightly differently, and that's what makes the difference. In animals, we can break down the alpha glucose linkages, but we cannot break down the beta glucose linkage. Okay, and again, it's the difference in how those are put together that leads to their different properties. So that butterfly's wings are made up of chitin, which is a structural molecular form of a polysaccharide made up of sugar molecules. Okay. So what we can see up here, the alpha glucoses is between the 1,4 carbon, and you can notice it looks very similar, whereas the beta glucoses are also between the 1,4 carbon, but they're positioned in a three-dimensional space slightly differently. And as you can see on the beta glucose, they are alternating, okay, in terms of where they go. Here they're all on the bottom, so to speak, and here we have one on the top, one on the bottom, one on the top. And what breaks these bonds are enzymes, which are a form of protein. And we as animals do not have those enzymes to break down the beta glucose linkages, such as wood. And you might think, well, what breaks down wood? And you might think termites. And you might think, oh, yeah, termites have it. Well, termites don't even have it. What they have is a symbiotic relationship with different types of single-celled organisms that live inside their guts that actually break it down. And same thing with cows and goats and all that. They're ruminants. They have bacteria in one of their fourth chambers of their stomach that actually break that down, because there's a whole bunch of bacteria and other types of organisms living in that can break those linkages. But the animals themselves cannot break those linkages. So once again, structure changes function. The enzymes that digest starch cannot break down. Hydrolyze means to break down. Hydro, water. Lyse means to break apart, so using water to break something down. The beta linkages in cellulose. So that's what passes through. When we eat plant foods, the cellulose does pass through our digestive tract, and that's what we call fiber, which is really important to help, clean your intestinal walls and stimulate that secretion of mucus. Okay, and as I say up here, microbes can digest that, and things like cows and termites do have a symbiotic relationship with these microbes. Okay, and as I said earlier, when we take in excess glucose, our body will store that excess glucose in the form of glycogen. Glycogen will be stored in your liver cells for the most part. That's where the majority of glycogen gets stored. And when our body glucose levels drop, then the liver will start to break down, hydrolyze that glycogen into glucose molecules once again in order to maintain homeostasis. That's the same level of our blood sugar levels. And many problems diabetics have is with the production of insulin, and glycogen is a storage form of sugar, and insulin is what actually creates the glycogen, because it helps to bring the sugar into the cells, and then it will, once in the cells, it can be either used immediately for energy or put into a storage form. Okay, it's highly branched, and we store it in our liver. And we're going to stop there, and that is the end of lecture 15-A, and 15-B will be about lipids.